Hello everyone, welcome back. My education is proceeding. Right now in my degree pre-studies, I've been doing histology and hematology. So I've passed on from virology, immunology, epidemiology, and that was really interesting. I learned a lot about, how viruses enter, how the immune system tries to deal with them, where vaccines come into the equation, how effective they are over multiple doses and between different demographics, calculating herd immunity, critical thresholds, all that kind of thing. I found it quite interesting. I learned a lot about immunoglobulins, aka antibodies, which was interesting for me because that's something I really wanted to get into, thinking about, my post-COVID breathing issue, my theory about molecular mimicry with dietary proteins. While learning about all this stuff as well, I did bump into some research papers where people are actually taking that molecular mimicry idea seriously, but not for dietary stuff. I keep bumping into even very recent research papers that are talking about COVID spike protein and capsid proteins, or the epitopes rather, having or manifesting pairs in terms of other structures in the body that the antibodies might mistake for being COVID. So this whole epitope pair thing is interesting. Epitope, by the way, is just like a pattern that the antibodies match, essentially. So the different antigenic structures, I'll try and not use the terminology, the different shapes of the proteins on the virus. Once your body kind of breaks it up and then has a look at it, it will find the appropriate cells in your body, B cells, which can produce things that match that pattern. It's just unfortunate that those things can also match other parts of your body, and that's what autoimmunity is. Auto, like self-immunity. So the thing that I would like researchers to look into, as we've stated in previous videos, is that I believe that there's a strong dietary mimicry component to people's COVID responses that's triggering mast cell disorders, leading to kind of systemic or cascading histamine releases, which is producing issues that people are defining generally as post-COVID syndromes. I'm unaware if there's been any research into kind of external proteins. Like obviously I'm ingesting milk protein and then I have a reaction to it. I believe it's because when it touches the mucosal layer, the epithelial cells there along the esophagus, that's when the reaction starts. And I believe that's releasing histamine and then oversensitizing the vagus nerve, which is giving me the the sensory breathing issue. But I don't know if there's been like a dietary epitope pairing study. I don't know how you would, it's probably going to be quite a difficult one to do. You'd have to track everything that people ingest over a period of time. But anyway, it's been interesting learning about it more. So the hematology is something that I've moved on to now, and I'm finding this one really interesting. So I've been looking at virtual samples. So we've got histology samples, basically just tissues from different parts of the body, learning about the different layers. The best way is to kind of planar, slice different samples where appropriate to get a good visual of the cell layers across a tissue. But then hematology is the one that I've really been interested in. A few months ago, I did a video cleaning out my microscope, and we had a look at a bit of my blood. And I didn't know anything at that time, really. I knew what blood cells generally looked like, but in that video, I was surprised because I could see the blood cells clearly immediately, and I thought, no, it can't be that easy, surely. Surely little old stupid me is incapable of doing something like that. What I've realized looking back is that, damn, that was actually a really good view of the erythrocytes, they call them red blood cells. Turns out my kind of bootleg microscope is actually not that bad. So I did it by pricking myself in my new favorite spot, and then just kind of pressing the microscope slide against it, which is effectively a blood smear. It was still a bit too thick, and you'd be able to see that in the video by the clumping of the erythrocytes. So it wasn't like a clean smear. But one of the things I really wanted to learn how to do was white blood cell counts or leukocyte counts. And now I know how to do them, but I don't have the proper staining tools or chemicals yet. But I now know how to identify all sorts of different things in an appropriately stained blood smear. So what I mean by that is, in the biomed field, there are all different types of stains you can use to, stain different tissue samples, or not just tissue samples, all sorts of samples. They are chemicals, sometimes potentially hazardous chemicals, but they kind of color code and highlight different types of tissues and cells. And then it goes into a whole other field of immunohistochemistry, which is a really clever thing of using specific antibodies to highlight specific proteins. So you can very selectively choose which molecules you want to highlight to make them easy to see under a microscope, which is so sci-fi. But anyway, for looking at red and white blood cells, the white blood cells are extremely difficult to distinguish without any staining. I don't know if it's impossible. I'm sure people have tried this, but I had this idea while reading about it. Surely there's some kind of clever light thing we can do. Nothing absorbs exactly the same wavelength. Surely there's a way to have different alternating wavelengths like projecting onto the microscope slide to kind of make it a bit easier to see the white blood cells. I'm sure that's got its own name and everything. I just haven't read about that yet. But anyway, the way to see the different types of white blood cells is to stain them properly. So I've been looking at samples and learning from them of properly stained ones using H&E stains, or what's it called, Giemsa staining? It's methylene blue and eosin, I believe. The reason why I want to learn to do that is because I've got lymphocytopenia and I would like to be able to track it over time. In theory, I would be able to tell if I have an infection, regardless of what the infection is, from here without needing to see a doctor or get a blood test or anything. That's the power of like doing this learning and building home tools to put it to the test. I also know how to detect leukemia. I'll be able to see whether I have blood cancer effectively. There are different types of leukemia. I don't know whether it's appropriate to say different types because I need to study leukemia more if I want to speak about that properly. So you have all these immature cells, I think they're called myeloid cells, that flood your bloodstream and you can definitely see them. And then there's another kind of category of it where the lymphocytes are kind of malformed and weird, but it's quite apparent when you know what you're looking for. We also had to look at malaria. So I know how to identify malaria. Malaria is a creepy ass one. So again, before doing all this learning, I didn't actually know what malaria really was. I mean, I knew it was like an epidemic mosquito type infection thing, but now I actually know what it is. It's a blood parasite, and it's so freaky and weird, like looking at it under the microscope, because you can see when stained these little marks, usually blue marks, depending on the stain, that represent the little parasites, like little granules of them in the red blood cells. And then over time, they progress to a point where you can see red blood cells that are quite packed with them. And then I believe they basically just explode at some point. But that's the cycle of how the parasite survives. Then a mosquito will come along, drink your blood, it will get in, it will transfer it to you via the saliva, I believe, as it's taking its little blood meal. Then it transfers the parasite back into you. So it's this thing which basically like replicates in your blood cells, not just in your blood, but inside your actual blood, which is freaky. But I now know what that looks like. So I don't have malaria, but in theory, I could do a little blood smear, have a look at the cells if they were stained, and be able to go, yes or no, this person does or does not have leukemia, yes or no, they do or don't have malaria. Then if I had done a differential white blood cell count over time, I could then do another one, compare it against the average and say, oh, this person looks like they're fighting off an infection or not. Then there are a whole bunch of other conditions relate to that. So I said for a while that one of the things I want to do next is upgrade my microscope with better digital camera capability, because the one I've got is not like a traditional brand microscope. There's no proper information on its specifications or any help for it. So what I've done is I've got a few adapters, which I think are turning up today for the trinocular scope. I'm going to see what fits and what doesn't. And I've got a 3D printer, so I can like build my own kind of adapting pieces if that's required. From there, now I don't know about regulations. I would like to go to a point where I can have little samples of the methylene blue and eosin stains, and I might need a fixative as well, because I would like to get to a point where I can do those white blood cell studies from here. That's like one of my big checkpoints for what I want to get to. But again, I don't know about regulations, and the thing is, if I do try and get hold of stuff that's a little bit questionable to get hold of, I do have a reason. You know, I'm a biomedical science student now. Well, I will officially be starting in October, so it will be an aid to the educational process. See, it's interesting. I mean, I've kind of been having this slow revelation over the past few months where, you know, I was just kind of cruising through life, doing my own thing, you know, in and out of all the ups and downs. But occasionally when thinking about things, you know, you just have these cursory thoughts about like science, medicine, the universe, physics, whatever. And there's this little thing in the back of your head that you don't even realize is there that goes, oh, that's for someone else. That's for someone else to do. That's for someone else to figure out. That's for someone else to learn, etc. It's weird now actually knowing how to do these things, knowing how to recognize these things, knowing what the things I've heard about for so long actually look like. And in theory, kind of how easy it was to learn that. Like once you know, you know. Like it doesn't take continuous effort. Like I'm not running on a treadmill to like earn this new knowledge. I sit down, I study, and now I know it. And it's just a weird thing that, you know, like a few months ago, knowing how to detect leukemia would have been like, oh, I'm sure that's something only experts can do. And now I'm like, wait, I actually understand it. I know what it looks like. I know there's a lot more I need to learn about technicalities, but now I know how to identify the markers for it. And that's what makes this fun. It's like you can actually like write down. I can write down if I wanted like a list of things that I have earned, I suppose, like new skills, cognitive skills. This is kind of exactly what I wanted, like building up this picture now over time that's going to be filled in with more and more detail, like actual real knowledge. Again, it's kind of interesting because we come from creative fields, right? And we can learn a lot about fictional lore, like video games, movies, etc., and get really in deep with that. Well, this character's motivation was this, that, or the other, or even like fictional magic systems and fictional scientific systems, and become obsessed with that. Even things like, alchemy in video games and spellcrafting and like ship construction and all that kind of thing. But it's like, oh wait, no, actually, this is real. Like, oh, I could just as well be reading a fictional book about, how some alien species works, but it's like, oh, no, wait, this is actually real. But I also think that's kind of a fun way of approaching it. Sometimes focusing on the stuff you're learning as real also kind of comes along with this side fault that it's boring or too important to mess up, and therefore it kind of makes it unattractive to engage with. Maybe it's kind of fun seeing the real stuff as slightly fictional, if that makes sense. I feel like that's also why I've been, like I said, for the first leg of my pre-learning, I was watching Jurassic Park a lot because it's got some somewhat biomedical elements to it, with all the DNA stuff. And it was more just like this immersive factor. I guess I kind of did it instinctively. I notice that now when I'm studying, I'm thinking, when I'm away from this space, I find it a bit less interesting to engage with. But when I'm here and I've got like scientific ambience, even sometimes, you see those videos, you got like ambience with faint dinosaur noises. I'm just focusing on Jurassic Park, but it doesn't have to be. But like, I build the space that makes it immersive, hopefully not in a cringe way, but it just makes it feel slightly less real. So it kind of feels like I'm studying something slightly fictional, but I think that really helps me to take it all in and makes it more fun. Also, I have different methods for learning. I kind of go through different phases and I make a shit ton of notes, as you can see. So that's exoplanet studies. That was beforehand. I also use, not controversial, let me explain. I use AI sometimes as a virtual teacher. What I mean is to generate me assignments to do in addition to my regular learning. But that's it. That's all I use it for when it comes to the educational studies. Just to give you like more points to think about so I can write myself essays. Sounds kind of nerdy, but there we go. Okay, so we've got what I call it, enhancement testing. So this is basically that. I generate myself assignments, additional assignments extra to the course, and then I write them. Helps me to solidify. I've got notes for the COVID-19 related immunology course. So that's a lot of notes there, which includes some diagrams as well, as you can see, talking about like viral structure and stuff. What have we got here? Spike protein. I do also wonder about like whether to sell this in like a little package to raise a bit more money for working on the lab. I don't know if anyone would want to buy my notes though. So yeah, look, I've basically written a book's worth of notes already. And then we're on to histology as well, and I sometimes have like little reference pages where I put important formulas and stuff on the back, and then here I've got like a little key for different like white blood cells there printed in color, so it kind of helps me to retest myself and kind of use it as a visual key. I've also started another couple of parallel courses to the histology, so there's one on antibiotic resistance, and then I'll do one on autism as well. So I'm trying to cram in as much as I can. All right, hello everyone. This is the first time recording from this specific computer setup. Let me just check this. There we go. So I'm actually in my little lab section, my little medical corner of the studio, and back there is the regular digital setup. You can see me on the screen because I'm editing a video. But I had accidentally just cut myself on the finger, and, as an aspiring biomedical student, I don't let anything go to waste. So I literally just smeared it on a microscope slide, and now, let's bring you over, hang on, and now we can take a look. So here are the erythrocytes, and it was interesting because, at least I'm pretty sure these are erythrocytes. I was literally just editing a video talking about my education, so maybe I'll throw this into it as well. So this should be them clumping together. Obviously I don't have any staining. What you're seeing on the screen, the user interface overlay, is something I was working on when setting up this computer, which was to have overlays. So that is technically a graticule, which I would use for determining scale if I calibrate it. And then also got a grid, hang on. So there's a grid, and the A marker is the dot in the center. So in theory, if I did do staining, and if I was doing like leukocyte counts, so differential leukocyte counts, then I would be able to use this custom grid to help with that. And then we'd scan along, and then we'd count and see which white blood cells were present. But obviously, like I mentioned, without staining, it's kind of impossible to tell like which of them are white blood cells and which are red. Another thing as well is I did have some adapter stuff arrive for the microscope. It didn't really fit the DSLR that I wanted to use, but what I noticed was that when comparing the camera kind of looking through either the trinocular scope or the eyepiece, it still didn't give the kind of clarity that I'm already getting from the integrated camera anyway. So I'm kind of thinking that the system I have already, that we're literally seeing on the screen here, is kind of better than I needed anyway. So I might just stick with what I've got. It is just a shame that I don't yet have the possibility for staining. Let's go up a scale, shall we? I've got food very soon, so I don't know if I've got much time. We're looking at 10 times magnification this time. I actually don't know what magnification the integrated camera is, but we can see how this looks. So I'm pretty sure these are the erythrocytes, but last time I saw these under a blood smear, I found it hard to believe until, a friend in the comments said that no, they were actually that. Let's go back to the higher scale. So this is 40 times magnification. There we go. I can do some adjustments on the lighting. I don't know if we'll be able to see any changes. So this would make it more Fresnel, as I'll call it, although that's not the exact terming, but when I've described it to my friend, I describe it as Fresnel. Again, you'll see that we only get the borders, so we can't see any nuclei in the cells, unless we can, but I'm just not that good at spotting. If I go here, no, I think the staining will be required because everything at this level is basically transparent anyway. And then we widen the amount of light, and it basically just looks like a three-dimensional blob. Can also decrease the amount of lighting, not that that helps us. Decrease while widened, no. Okay, for something like under the graticule and the center point here, maybe that's just like a regular erythrocyte because we see the dip in the center of the lighting. But what I'm basically discovering is that I really like this microscope. It was kind of just like it wasn't cheap. I don't know if it was like £300 to £500 or something pounds on Amazon. It's from a company called Vabiuf. It's basically just like some, Chinese thing, whatever it is. But I've become a bit more happy about it, I suppose. Maybe I'll cut the recording here in a sec. But so this is just what I'm dealing with prior to any staining technology, which I would like to get my hands on if regulations allow. I could have a section in the studio for preparing the microscope slides. That has clotted up now on my finger. So yeah, interesting. I just thought, you might find it cool to see what I'm doing here in the corner. So more capability will be added over time. Oh, obviously, obviously you haven't seen this yet. I have a centrifuge as well in my little lab space, so I wanted to get some pots that were compatible with it. So that's what I've done. What would I use this for? Well, I'm not drawing my own blood, in the near term at least. So maybe for biological samples. I live in an area where we've got quite a large diversity of kind of ecological zones, if you want to think about it that way. So I was thinking maybe it'd be interesting to get into the biology, maybe record some of it, then maybe use some of those recordings for artistic purposes, or maybe do some actual science with it. All right, so for now, I'll leave you be, and I'll get back to finishing that studio video. And then I've got a whole bunch of stuff to do on the main channel as well. And I've probably already done an outro on that. Maybe I'll use this. If you made it to the end of this video, please do a microscope, a lab thing, or an education thing. Obviously, have a great day. You can support me on Patreon. The Patreon, by the way, contributes to all this stuff. Have a great day, and I will see you next time.